The overall goal of this procedure is to isolate airway epithelial cells in order to study the toxic effects of cigarette smoke exposure. This is accomplished by first dissecting mouse tracheas and then digesting the tracheas in pronase solution. The second step of the procedure is to collect epithelial cells and propagate them in liquid culture. The third step is to differentiate the cultures at an air-liquid interface. The final step of the procedure is to expose epithelial cells to apical cigarette smoke at the air-liquid interface. Ultimately, results can be obtained that show a fully differentiated monolayer of epithelial cells by immunofluorescence microscopy.
This method can help answer key questions in the chronic obstructive pulmonary disease field, such as the effects of cigarette smoke exposure on cilia morphology and cigarette smoke-induced biochemical markers of cell injury.
Begin preparations for this procedure 1 day prior to the dissection by preparing Ham's F12 media containing antibiotics, Ham's F12 media containing antibiotics and 20% FBS, MTEC basic medium containing antibiotics, MTEC medium with 5% FBS, DNase 1 solution, retinoic acid stock solutions, and MTEC proliferation medium according to the instructions in the written protocol. Sterilize all surgical tools to be used in the dissection. On the day of the dissection, ensure that the dissection area is clean and that all dissection tools are at hand.